Welcome to the Cocrystal Pharma presentation. I'm Gary Wilcox, Chairman and CEO, and I have with me today our CFO, James, and I have Sam, our President. And we are very pleased to have this opportunity. The opportunity that we're pursuing in the company is the curing of viral diseases. And arguably, viral diseases are amongst the most serious, most intractable problems of humanity. And while we've witnessed pandemics in the past, there's never been the attention paid to them that is being paid to the present pandemic with COVID-19. And I think we often forget that these viruses are able to mutate, and when we do get drugs against them, the mutations will arise in the viral population, and as a result of that, resistance to therapeutic drugs and vaccines can arise in those populations. And so this isn't a one-time fix for COVID-19. The virus will evolve, and new medicines will have to be developed, and new vaccines will have to be developed. And we can just look back at the AIDS pandemic that started back in the early 1980s, and over 40 million people have died from AIDS. So this isn't the first time in recent times that a very serious and initially novel and intractable virus has arisen and caused tremendous personal loss in terms of disease and deaths, and great economic burden. So if we can have the next slide, I have to talk about some forward-looking statements. And so I'll be saying things like could and may and will and potential. And you have to recognize these are forward-looking statements. On the next slide is just a quick overview of Cocrystal. We're a clinical stage antiviral company. We have a very powerful proprietary drug discovery platform that'll be described during the presentation. We have an exciting collaboration with Merck, and just had a press release on that earlier today. And we believe that we have potential for developing prophylactic as well as therapeutic drugs. And the viral diseases we look at are influenza, COVID-19, hepatitis C, and norovirus, which causes a severe gastroenteritis. On the next slide is our investment hypothesis. And we're applying a structure-based drug design technology that we believe will develop first and best-in-class broad-spectrum antiviral drugs. These are very large market opportunities. The treatment of acute and chronic viral diseases, including both seasonal and pandemic, influenza, COVID-19, hepatitis C, and norovirus. In each program that we have, we test products for multiple ways of delivering them to patients. It can be in a hospital setting, IV, it can be IM, it can also be a vaccine in the general population, and it can be a therapeutic that could be given in the form of a nasal spray or a product that's delivered directly into the lung or a pill that's swallowed. Robust development pipeline includes a Merck collaboration, and we'll talk more about that later on in the program. We have a seasoned leadership team that's composed of 2 Nobel Prize winners. One, a co-founder of the company, Dr. Roger Kornberg at Stanford University. We have a scientific advisory board that Roger shares that's composed of people who are world experts in their fields, including another Nobel laureate, Dr. Michael Levitt at Stanford University, And major parts of our technology platform, which of course include computation, fall under his area of expertise. On the management team, we have Sam Lee, our director and head of research, and our president. Sam trained at Stanford University, and prior to being at Cocrystal, was at ICOS Corporation, where he led a very important team that developed what became eventually a marketed drug called Zytolic. I was also at ICOS where Sam and I first met, and had the opportunity to play a key role in the development of Cialis. Jim Martin, our CFO, has had 25 years of financial and management experience in the healthcare industry, and especially in publicly traded companies. So Jim has the ideal background for being the CFO of a healthcare company like ours that's a public company. And of course, we're traded on NASDAQ. On the next slide is the description of our technology drug discovery platform. And our focus is in the area of preventing viral replication. When a virus infects us, it enters into cells. Inside those cells, it can replicate itself. And if you can stop the replication process, you can halt the viral infection. It begins with selecting a target, and those targets are protein in nature. We can make those proteins in a variety of different cell types. We can purify them, we can characterize them, and very importantly, we can make them into crystals. And those crystals are what we use to determine at the atomic level the structure of our protein and our target molecule. And those structures are key for going forward, because by being able to see each atom, we can identify areas where we'd like to find a particular drug binding, and then we can start screening compounds to identify compounds that inhibit the replication activity of our purified protein. We can then take that compound and complex it with our crystal and end up with a cocrystal. And now when we determine its structure using high throughput X-rays, we're able to see where our candidate actually binds, and then we can use that information to make more potent and more drug-like molecules. And then we go back again to the X-rays. The X-rays that we use are available on contract from either the University of California or Stanford University in multi-billion-dollar complexes that exist on both of those college campuses. And we can use those to continue to iterate this program. So we have a fully optimized operation that goes from expression of the protein target all the way through high resolution X-ray crystallography. Once we have a good idea of what's going on, we iterate the process and produce better and better drug candidates, and eventually take those into preclinical studies, and the goal being to get into the clinic and see that they're both safe as well as effective. So on the next slide, I'll show just quickly here our pipeline that we'll be discussing during the rest of the presentation. And that pipeline has two programs in influenza. One is in collaboration with Merck. We also have a very important program in Covid19 with a replication inhibitor, and a second program with a protease inhibitor. We have a pandemic influenza A program, which is 100% owned by Cocrystal, and we're focused there on the strains that can mutate from the present circulating strains of influenza A and become a pandemic. And influenza is another terrible disease. It's a respiratory disease just like COVID-19, and some of the same sequelae to infection that we see in COVID-19 resembles what are seen in influenza infections, but there are very significant differences as well. We have a hepatitis C program that's completed phase 2A studies, and finally we have a norovirus program. So let's move on and take a look at our program with Merck, which is shown in the next slide. It begins with making crystals of various strains of influenza A and influenza B viruses, as well as existing drug-resistant mutants of both influenza A and influenza B. We look for very broad spectrum activity, and we also look for ways to design the drug so that it will be resistant to future mutants that we can actually predict. Why can we predict them? We can predict them because we know the structure not only of the target, but the structure of the target with our drug candidates bound to it. So if we go to the next slide, we'll look at the commercial terms of that relationship. We have an exclusive worldwide license, and the first part of the agreement was a collaboration to discover and develop proprietary influenza AB antiviral agents. So over the past 2 years, Cocrystal and Merck have jointly developed some potent influenza AB inhibitors. All of Cocrystal's expenses were reimbursed by Merck, and very importantly, Cocrystal met all research collaboration agreement obligations under the agreement. In the next phase, Merck will be responsible for continued R&D, including clinical development and worldwide commercialization of products that derive from the collaboration. And Merck is responsible for funding all of those. Cocrystal is eligible to receive from Merck up to $156 million in milestone payments as the program at Merck progresses, and also will be receiving royalties on product sales. We can go to the next slide. I'll now describe our COVID programs. These come, as shown on the next slide, from Kansas State University. There was a remarkable group of people at Kansas State that Sam Lee identified and got to know quite well over time in the veterinary school there. And they were studying coronaviruses that infect cats and cause some pretty serious diseases in cats. And it turned out that the compounds that they were studying also have antiviral activity against the SARS-CoV-2 virus, which is the virus that causes COVID-19. And there was data showing that there was activity in proof of concept animal models. So this was a very exciting opportunity for us because it allowed us to basically license in a program that had been in development for many years. And in fact, these groups had worked for over a decade on the coronaviruses. So we have exclusive patent rights and know-how for both coronaviruses as well as noroviruses for therapeutic use in humans. On the next slide, what we have is just a simple statement about COVID-19, and that is that at the present time there's no approved antiviral prophylactic treatment, and there's no effective COVID-19 antiviral therapeutic treatment. And this will be very important as we go forward, because as wonderful as the vaccines are, and I believe they will be used effectively for a long time. Vaccines usually don't completely rid a population of the disease. They reduce it. They may ameliorate the clinical manifestations of the disease, but the disease usually hangs on. And influenza, of course, is an example of that. If we look at the next slide, what we see here is very important. It's the time course of an infection with COVID-19. And initially, if we look at the black line that goes through there, you have infection, and the sole response is replication of the virus in certain cells, mainly located in the lung. Then, after a week or so, there's progression to a second stage in which the host inflammatory response really kicks into gear in response to the viral replication that's taking place. And after that phase 2, it goes into stage 3, and there the host inflammatory response increases very significantly. It can go into a hyperinflammatory phase, and that is very, very serious, and that's what can lead to death. So if you go back to the left side of that chart, you can see that stopping the viral response in stage 1 would prevent the host inflammatory response that occurs in stages 2 and stage 3. And so all of our work is focused on inhibiting the replication, and that would be important mostly in phase 1, but also to some extent during phase 2. We can have the next slide. What we show here is that we've had an achievement of a number of milestones during 2020. After obtaining the license, we filed additional patent applications. We've done some additional proof of concept animal model studies, initiated preclinical studies on the COVID-19 inhibitors, all that in the second quarter. In the third quarter, the identification of additional inhibitors using our proprietary technology to give us additional opportunities for potential drugs. And in the fourth quarter, we announced that we've selected a preclinical lead from among the KSERF, Kansas State University Research Foundation licensed inhibitors. If we can have the next slide. I'll now move to the antiviral program that we have on pandemic influenza, and that's the A strain. And there's a really significant need to be ready for influenza strains that become pandemic strains, because we know from past experience that it's already occurred and it will occur again. The number of cases annually in the world are estimated to be over 1 billion. That's 1 in every 7 or 8 people in the world becoming infected each year by the influenza virus. 3 to 5 million of these cases are severe, and up to 650,000 deaths worldwide on an annual basis. The challenges that face influenza are that current antiviral treatments will work for a while, but then viral resistance appears due to mutations in the viral genetic information. And as a result of that, this resistant population makes the approved therapeutics that are presently used to treat influenza ineffective. And the two main ones, Tamiflu, has a long history of drug resistance issues. And more recently, Xofluza, which was approved in November of 2018, surprisingly showed the emergence of drug resistance mutations within several months of the time it was first launched. And that was a surprise. And even more of a surprise was the observation that the emergent drug resistance was already present in the population. So in the US, as many as 10% of strains have been resistant, and in Japan, even a larger number, close to 20%. So that's a problem that needs to be addressed with any new therapeutic drug. And if we can have the next slide, what's shown on this slide is that flu is a serious problem, and that it's topped 80,000 people dying from it in the United States in the 2017-2018 influenza season. If we go to the next slide, what we see here is our approach. Again, it's to make crystals from the replication enzymes of the influenza virus from a broad variety of strains, including existing pandemic strains, as well as drug-resistant strains. And that's what's shown in the pictures across the top of the slide. What we've done is to use the platform technology that we've developed. We've been able to identify compounds that have a very favorable preclinical safety profile, that have good pharmacokinetic properties, and that could be potentially administered by different routes, including oral, inhalation, and injection. In 2020, in the second quarter, we secured our supply line for materials to make our lead compound. In the third quarter, initiated our second batch of API synthesis. And as a goal this year, in the third quarter, we want to be able to initiate a phase 1 study. So if we can go to the next slide, what we show here is a very important property of our preclinical lead compound. And that is, if we take approved compounds, such as the two I mentioned previously, Xofluza and Tamiflu, and ask what happens if we have a combination of either one of those with our preclinical lead compound, is the effect additive or is the effect synergistic? And the answer is that there's a strong synergenic effect. And that synergistic effect is basically that 2 and 2 is 7 or 8, it's not 4. And so this is an important observation because it opens up the possibility of using a cocktail to treat influenza. And if one looks at viral diseases in general, AIDS is a good example, hepatitis C is a good example. The most successful therapies that have been developed are cocktails, combinations of 2 or more drugs. Let's go to the next slide. And what we have here is the long list. It's not the complete list, but it's a list to show that we've taken our lead compound 42344 a long ways through the pharmacological safety and toxicological tests that need to be done prior to entering into the clinic. And we have a few more to do, but we're a long ways down the road to where we have to be. And the green check marks there indicate that the compound has passed each one of those hurdles. All right, let's go to the next slide and talk about our hepatitis C program. And here we have a different kind of marketplace into which we are entering. And what we believe is important in hepatitis C is to have a short treatment regimen. There are good drugs available today. AbbVie's Mavyret, Gilead's Epclusa are excellent drugs, But Mavorite is 8 weeks of treatment, Epclusa is 12 weeks of treatment, and longer periods of treatment lead to more time for the virus to replicate and mutate, and that can create drug resistance challenges. In addition, of course, the longer you treat with any drug, the greater the increased risk of an adverse event occurring. And here, the opportunity for missed doses is critical because these viruses replicate very, very rapidly, and a missed dose can really reduce the efficacy of the drug. So we believe that our compound, 31244, which has completed a phase 1A, 1B, and 2A studies, is now ready to become part of a cocktail, a combination with already approved drugs or drugs about to be approved to develop new and novel therapeutics for hepatitis C. So in the next slide, what we see are the results from our work that indicates that we have a mechanism of action which is called a non-nucleoside inhibitor. It really doesn't matter too much what that is, other than to say that it's a long name for a mechanism of action that distinguishes our compound as belonging to a certain class of drugs that people have long looked at to try and find good drug candidates. And if we look at the table on the right side there, you can see that there's a compound ABT333 from Abbott and GS9190 from Gilead, and both of those are effective against genotype 1. On the other hand, our compound, 31244, is effective against all genotypes of the hepatitis C virus. So broad spectrum. In the far right column is the viral load reduction. And our compound reduces 1,000-fold the number of viruses present in patients undergoing treatment, whereas the other two compounds have a reduction of less than 10-fold. So this drug is orally administered, it targets the liver, and it's ready for combination therapy in clinical trials. And if we can have the next slide, our 2A trial was 6 weeks of Epclusa therapy, including 2 weeks of 31244. Treatment was well tolerated. There were no study discontinuations. 8 of the 12 patients achieved what's considered to be a virologic cure. That is, there's no detectable virus after 12 or 24 weeks from the time that treatment stopped. However, these 4 patients that had a virological relapse were of concern to us until a lot of work was done to look at the blood samples from these patients. And what was discovered is that the 4 patients that failed had a significantly higher frequency of a class of T cells, white blood cells, called CD8 positive cells, compared to the 4 who had relapsed. So very important observation there that provides an opportunity for personalized medicine in a routine screen before a patient would go on to a combination therapy. One would want to know that they have a good supply of CD8 positive T cells because those are the patients you'd want to treat with our compound 31244 as part of the cocktail. So our next steps there, in the first quarter of last year, we filed this report on the phase 2A study with the US FDA, and we've now achieved what we think is as far as we should carry this program forward, and we're seeking a partner here for further development. Right now, the next slide, what we're looking at is the final program, norovirus gastroenteritis, and on the next slide we can see the approach that is very familiar, where we isolate different strains of the virus. We pick a target. We purify the protein target. We make crystals. We determine the atomic structure using X-ray crystallography. And from there, we take initial compounds that inhibit the target and further develop those into preclinical drug candidates. The norovirus is best known probably because of the effect it has on cruise ships, where they have to return to port whenever there's an outbreak. The estimated annual cost of norovirus infections on a worldwide basis is $60 billion. 385 million infections worldwide annually, about 20 million cases in the United States, about 465,000 emergency room visits, and 109,000 hospitalizations. These are all numbers from the CDC. So it's a significant disease. It's one for which there's no approved therapy. So let's look at the next slide. And we believe we have the potential, and we're on the road to developing the first therapy for norovirus. We've isolated good compounds. They're potent. They have broad spectrum. They're active against two targets. The first one is the polymerase enzyme that replicates the viral genetic material. And the second one target is a protease, and the protease cleaves an inactive form of the polymerase into the active form. And so if you inhibit the protease, you don't get the active form of the polymerase, and the virus is not able to replicate. So we're at the structure-based lead molecule discovery stage. In the second quarter of last year, we filed additional patent applications. And in the first half of this year, we intend to complete proof of concept animal study. So this really concludes the description of each of our programs. And now I'd like to turn the discussion over to Jim, who's going to talk about why he feels we are so well positioned for growth and talk about our financial position.
Thank you, Gary. So I'll start off with our IP portfolio, because this is very important to us. Every program that Gary just went through, we want to make sure is well protected. As you saw earlier, we have a management team that's experienced in having success, and with that success comes challenges on your compounds and on your drugs, and trying to work around to find a way to take away some of your your gains that you'll get from these winners. And so with that, we build a very strong patent portfolio wall around each one of these early on, so that when they are successful, we are protected. Our financial snapshot, we have around $100 million market cap. We have a good volume in the stock. Our balance sheet is very clean. We have over 30 million in cash available. Primarily all we have is common stock. There's no preferred stock on our balance sheet. We also have no debt. So it's a very clean balance sheet. We have a very nice burn rate that will have our cash runway going out beyond 2022. We can show this slide again. This is our investment highlights. Just quickly going through these. Again, we are applying our proprietary drug discovery platform for first-in-class and best-in-class compounds that Gary had went over earlier. This is a proven platform. We've already had clinical data coming out of drugs off of this platform. This is the same platform that is used with Merck to create the compound. So it's a proven platform. And also you've seen all of our programs, both in influenza, COVID, hepatitis C, and our early stage neuro, and these all have large markets and great needs. I also think we highlighted how when we do develop, we make them broad spectrum, and we also make sure we have multiple routes of administration, because if you have an influenza therapy, some people may need it in a oral form, some people may need it, by IV, depending on their situation. And we also highlighted our Merck, which we just put out a PR this morning. So you can see that that program is moving along fantastically. Strong leadership here. We have 2 Nobel laureates, lots of experience in the management team with successful companies, starting off where, developing drugs and going into commercialization. Aside from that, I just went over the strong cash position that we have and how we are a very lean and cost-efficient company. We don't overspend. We're very thoughtful on that. And I think we have a nice runway. And with that, I'll conclude and just say thank you.
Gary, Jim, thank you so much for your presentation. I'll take over for the Q&A session.
Thank you. Hi, Ahu.
Hi, nice to see you, Ahu.
Likewise, Gary. Thanks for the great overview. So we have only a few minutes, and I received a couple questions from the investors or other audience. Majority of the questions are focused on the coronavirus program. So one of the questions we receive is, are you looking for a partnership?
Yes, we're not actively looking for a partnership at this point in time. We're opportunists first and foremost, Ahu. And so we believe that we're adding very significant value at this point in time until we get to the point where we know we're in the clinic or have entered the clinic. So as a result of that, we could end up doing a partnership earlier than I might think we would. If something came up, we'd take the opportunity, of course. On the other hand, we're in a very important position now, and that's why we've raised the cash that we have. I don't ever like to be in a position where you have to do a licensing deal as a way to raise money for the company. That's not a good position to be in. And so we raised the money that we did. We have over 30 million, as Jim just mentioned. And that really puts us in a very favorable position to ask the question every day, should we keep going on the path we're on? And can we go on the path we're on? And yes, we can. We have both the technical knowledge as well as the capital to do it. And of course, we would consider the right kind of partnership and the right kind of partner. And I think we've also demonstrated to our shareholders that we're capable of doing that because of the wonderful relationship with Merck, the results of that relationship, and now one of the finest companies in the world in the infectious disease area, carrying forward the work that we initiated.
Thanks, Gary.
If it's okay, I'm just going to add, yes, we are adding value right now to these compounds. And I just want to reemphasize that although there's been a lot of news in the vaccine markets, especially around COVID, there isn't much news in therapeutics. And that's where, that's coming now. You're going to need therapeutics to support vaccines the same way there's vaccines for influenza and still people catch influenza. So, and what we're doing right now is really going to add value, and we're not going to sell ourselves short by, licensing too early.
That's very helpful, Jim. So we have multiple other questions, but we don't unfortunately have any time. Congrats on the Merck collaboration that was disclosed this morning with the new developments, and I think you are positioned really well with your capital in hand and in addition to your diverse portfolio. So congratulations and thanks for your participation.
Thank you, and thank Noble.
Thank you very much.